Hi, you guys. My name is Dave Holmes. I'm founder of Clade 9. We started as a consulting company for cultivation. Came out of LA. I think we formed in 2009 and then kind of scaled up our operations in consulting, mainly for dispensaries in the LA area. We were managing cultivation operations. And you know how LA is, it was very unregulated. So in 2013 we had some incident and we had to kind of change our path. I was lucky enough to go and meet a group in Phoenix, Arizona. And we kind of took the consulting model to the regulated market. And that turned out really well for us. So anyway, I was able to go to Phoenix and launch a really large-scale operation. It was a lot different than California on the cultivation side, obviously, because it was completely regulated, seed-to-sale tracking. There wasn't lab testing yet, but it kind of really got me thinking about the regulated market. And so that experience we took to Vegas, and we used that to launch an operation there. Anyone been to Nevada lately? To Vegas to buy any recreational cannabis? Well, it's a very good market. And the operation we launched there was called Virtue Las Vegas. And that was probably our first experience in a very regulated market where there wasn't only seed to sale tracking, there was pesticide testing, testing for molds, testing for mycotoxins, heavy metals, very similar to what California is now essentially launching. So taking that experience, bringing it back to California, today we had just applied for cultivation, extraction, distribution in City of Los Angeles Phase 2. Anyone applied today in Phase 2? Okay. That's why you're here. It was very stressful. So anyway, that's our background. And kind of within the consulting and the cultivation operations, we develop breeding programs for our clients and also internally for our own operations. And we also developed a plant cannabis-specific plant nutrient that we also sell as a separate business. So thank you for having me.
Hello. Just be sure to stay on. My name is Stefan Kirchanski, and I'm a patent attorney. I've been doing this for about 25 years now, maybe a little bit more. As my wife says, this is actually my third career, and I'm not allowed to have any more, so I guess I have to make the most of this. I was originally an academic botanist. I got a Ph.D. at Berkeley several centuries ago, and my plan was to do academics. I actually got a job at UT Austin, which was a good botany department. And I liked teaching, I liked the students, and I hated the administration and all the bullshit that goes along with it. So I kind of bailed out of that, and I went and postdoced at Harvard for a while. And the lab that I wound up in, even though I was really interested in chloroplast membranes, I wound up in a blood lab because in those days people used red blood cell membranes as the model system. So when you can only kind of coast on something nice like that for so many years, and when I finally had to get a job, I just on a fluke I applied to a company, and it was actually a blood company, a blood counting company, medical company. And since I came from a blood lab, they figured that he must know something about blood. So they hired a botanist, and I learned a lot about blood and also chemistry and fluorescent antibodies and all sorts of good stuff like that. And when that finally dried up, my wife had gotten a tenured position at UCLA, so I went to law school and was doing sort of standard patent stuff all this time. The last couple of years I've been working at Zuber, Lawler, Duca, and they decided that they were going to focus on cannabis. And I had done a number of plant patents. I knew obviously not on cannabis at that point, but I knew about that, and I have since done cannabis plant patents and utility patents. That's the story.
I'm Reggie Gaudino. I'm the Chief Science Officer tester for Steeple Labs. Steeple is the first cannabis testing laboratory probably on the planet, at least commercially. I'm also a patent agent. I'm not sure how I ended up in the cannabis industry. I've done a lot of different things. But I seem to either piss off regulators the most or clients. We can't keep either of them from yelling at us. But basically the reason for that is we have kind of been the innovator in a lot of the testing that annoys people. Like we were the first to do microbial testing, RSA testing, pesticide testing in cannabis. We're also one of the centers for cannabis genetics research. We're partnered with the University of Colorado in their cannabis genome research program. We've published a number of papers with them. It's funny that the company's name is Clade 9 because that's just about how many clades we've discovered in cannabis for different things. Based on properties. So it's funny that you picked the correct name. So that's kind of where I'm from, Steep Hill. We were the ones who identified the potential cannabis patents that were disruptive to the industry. We've had a genetic screening and breeding markers to the breeding program for clients, and we've been kind of trying to push the industry down the pathway of patenting because that makes sense where I come from, which is plant biotech. But that seems to be another, I guess, one of the reasons why we're here today. So.
Great, thank you for those introductions. So to kind of start off, I think the conversation surrounding intellectual property and strains and all that is really relevant. A lot of cultivators are dealing with the hassles of, or not the hassles, but claiming they developed the strain when really there was a breeder before them. So could we start by kind of just discussing, explain the difference between like what a trademark and cultivar is in the realm of breeding?
I think the real problem there stems really from the intersection between patent law and trademark law. And I think you guys all understand that trademarks you think of as brands, and they really serve a dual function. The person that owns the trademark is developing goodwill in the trademark. It's a known mark, and they're protected from somebody else ripping their mark off. Originally, and still, one of the underlying justifications or reasons for trademarks is the public. It protects the public. They know what they're getting, and that it's legitimate, it's not a knockoff, and they know what to expect. Fast forward to the other side, we have this thing called plant patents in the US, and this allows people to have protection for clonally asexually reproduced plants. And it was originally invented to protect largely fruit trees and horticultural things. And early on, and this also comes up in utility patents, the problem is that if you have a trade name for something, that's your trademark, it better not find its way into any kind of a patent, because a patent is supposed to use the generic things in a trademark, or it's supposed to use the actual ingredients in a patent. So you have to know what something is separate from a trademark, because if it's a trademark, you might diddle it or change your product, and yet the patent is talking about something under that name, and it gets all screwed up. So it's been traditional, well, it's actually not even traditional, it's necessary to avoid a lot of problems. In a patent, either a utility patent or a plant patent, whatever you're talking about, if it's a process, or in the plant patent, if it's a variety, you give it a trick name, an internal name, like most plant patents, like rose patents, go under Cords 347, or Gen 1, for a guy named Jensen that did a lot of rose patents out of the Netherlands. That way, that's the name that the government uses, and the trademark that you sell the product under is completely separate, so that when the patent lapses, the trademark doesn't die also. If you keep referring to something in a patent by a trademark type name, the whole way patents are constructed, when the patent lapses, your trademark could very well lapse also. So you've got to be sure that you keep these things separate.
In many cases, actually, during the prosecution, they'll actually tell you you can't use that name, change it to something generic.
Right, the good examiners will tell you that. Occasionally, you'll see things sneaking through. the first cannabis patent that came out, I can't even remember the name of that thing now, but it has obviously a very commercial sort of a name on it. And I don't know if they're actually selling that cultivar. You know, I call it cultivar, you call it strain. It's kind of like corn and maize, right? But the first one that issued had funding. If you look, there's a bunch of them in the pipelines that are going along. Some of them are up again, down again. There's a bunch that have come out of Israel, and they've sort of done it the right way. They've come up with funny names that are not going to be a commercial name. If you ever see it in line, which I hope will come out pretty soon now, I have a funny name. I don't use it anymore to market it. Although, to keep my clients happy, the name that they're using, you could kind of figure it out from my patent name. Like I use some initials that helps them to remember which brand it goes to. Otherwise, they get confused too.
Perfect, thank you. So continuing on the IP and the breeding, Dave, you've been around for some time now, and you've seen a lot of cultivators go back and forth who really created the original strain. What steps kind of have you taken for the strains you guys are breeding for recreational applications to kind of protect it and really just let the public know that this is a production of Clad 9 versus another cultivator that might be making the claim that they created the strain?
Yeah, it's pretty tricky, you know, especially with, you know, the black market being pretty much the rule of the land until very recently. So, you know, before that it was just like a lot of sharing genetics. As we got closer to kind of legalization and at least in terms of states, we started to, you know, be, I mean, just, you know, we had to, by necessity, be less generous with the genetics. So we started to write contracts. We actually do have a strain patent in the pipeline also. So, you know, that's, I think in our future strategy is really to patent as many varieties that we have, cultivars that we can. But in terms of preserving the name, you know, we just try to establish our reputation and associate the genetics that we develop with our name. But that was the deal. Old school today, it's strain patents, plant patents.
So there's actually a right way and a wrong way to go about this. So one of the problems traditionally with the industry has been that, you know, we go pheno hunting and then we find something, we start making cuts, and then we start distributing to our friends. Dude, this is an awesome cut, you need to know this, right? So the problem with that is that now you've lost control of the genetics and it's out there everywhere because your buddy's giving it out, their buddies are giving it out, and suddenly everybody around the world's got that particular pheno. What has to happen in the industry realistically is an understanding of what plant big ag looks like, and then as much as people hate to hear this, is to start to model themselves after that, right? That's the right business way to do things for plants. The reason being, you know, if you have worked hard enough to find a unique phenotype, you need to stabilize it and you need to control people's access to it. Otherwise, you lose the ability to exercise all your rights, patent rights, anything else, right? So, and so a patent gets you so far, a trademark gets you so far, but you still have to have the proper business acumen to be able to control that so that you ultimately have to be able to determine where it goes. So this gets back to who was the original breeder? Well, if the original breeder had done a better job and could really trust the people or had handed it out on a license from the very beginning, he wouldn't be trying to verify the fact that he was the original breeder, right? So a lot of what we did underground, because we had to as an industry underground, has to change, right? And things like patents are not a bad thing if it's done the right way, and things like patents can help you build a portfolio and get people to look at you seriously for investment if you have that paperwork but that paperwork has to be predicated on the fact that you control access, you stabilize strains, and now you can talk about the next thing here, which is the genetic analysis and genetic influence, right? How do those things make your plant different so that you can say, look, I have this, it's different because of this, and that's what I'm getting a patent on, because ultimately a patent is not just for anything under the sun, it has to be for something unique and novel that can be verified as being different than something else.
And you sort of alluded to it, but being a patent attorney, I have to say this again. Besides the fact that you lose control, there's something in the US called the on-sale bar. If you sell something, or if it's publicly available, you have 1 year to apply for the patent. So if you have a new strain, give it to somebody to test it, you're already not only lost control, but you may very well have screwed up your chances of getting a patent. They're not, obviously the patent office can't check all those things, they don't check those things, but if you did get a patent, and if you ever had to try to enforce it, let me assure you, the lawyers on the other side are going to find out and shoot you down for that. Plus, of course, you know that there are all these public genetic projects now that are trying to take all the old strains and put them online to act as prior art for the patent office. So once you let something out the back door, if you haven't applied for a patent, forget it. You may not want to apply for a patent. I'm not saying you should apply for a patent for every single thing. Be careful.
So it's important to understand. So the way the law is written is it's offer for public sale. Offer.
You're going to have to sell it.
Right, exactly. You don't even have to complete a contract. If you make an offer for public sale, if you say, hey Joe, I'll sell you 50 of my newest thing, that clock for that 1 year starts ticking on that day. If anybody else heard it, it was in a public forum, that day is your day one. Right?
Our office will find that.
What?
They look in grow forums, they look everywhere.
Yeah, they've gotten smarter. It used to be when of course, when I first started this thing, we didn't even have the internet. But even after the internet got going, until fairly recently, the PTO wasn't very good at looking at stuff. Then they got smarter. They started looking at the scientific literature, which is usually quite helpful. But now in the cannabis space, they're looking at all of the common stuff on the net. And with the Wayback Machine and all these people that are archiving nets, they can show exactly when something popped up, and that can shoot you down.
Perfect, thank you for that clarification. So kind of continuing on that, Reggie, could you speak to the work that Steve Hill and yourself are doing when it comes to genetic analysis and kind of how that is really changing the future of the cannabis industry?
So, we all know that cannabis is a really great plant, right? So, but why is it a great plant? So we know about the cannabinoids, we know about a few cannabinoids, there are 144 cannabinoids, there are over somewhere between 250 and 500 terpenes that can exist in various strains, of which anywhere from 80 to 120 will be made in any given strain at any given time. So when you start, and then there's flavonoids, there's isoflavonoids, there's all sterols, there's all these other things that we know are biologically active from other plants, right? So now the question is what makes those things tick? You know, what are the genes behind them, and how do the different strains produce those things differently? Is there a presence, absence? Do some of them make some things and some of them don't? Are there differences in expression? Are the ratios between those things different? So the work that we've been doing is really looking at that. We've done a tremendous amount of DNA sequencing and RNA expression analysis. And some of those things we've actually put out in the market. We've already got, we've had a CBD test out there for a couple of years. We've helped MTG Seeds and Coastal Genetics and, build germplasm. So we have, I come from plant biotech. So the way I look at things is, we need to understand the plant and we need to be able to breed for the things that we want to breed for, because there are things in the plant that we're interested in. So what we've done is build a marker-assisted breeding program. We've got consulting programs, but the bottom line is to be able to use the tools that they use in big ag to be able to look like big ag in the cannabis industry, right? Because anybody who thinks that Monsanto and Dow and those people are not coming to this industry are kind of putting their head in the sand. So in order to be able to stay relevant and to be able to keep yourselves from being swallowed up, the right way to go about this is to breed your balls off and file patents on everything, right? So, and how do you do that is to use those advanced techniques, marker-assisted breeding. If you're looking to make THCV, can you find a gene that allows you to track that and then breed for that so you can augment it? So that's kind of what we do. We're not the only ones who do it. I will say though that we have kind of a competitive advantage because we're the only genetics company that's also a full-scale chemistry company, right? So we're the only ones that do both genetics and chemistry in the industry. And so because of it, we have a lot of insight into the genes that actually are responsible for the phenotypes that we're interested in. We came out a few years back with something called GenKit, which was an early sex testing. Other people had tried to do it, but it's not an easy thing, and hermaphrodites happen, and that screws things up. And so we're looking at hermaphrodite markers, we're looking at stress response, believe it or not. If you water your plant with 50-degree water in a 78-degree room, you hit stress it every freaking day. And that's going to increase the propensity to hermaphrodite. All these things matter, right? So, and so what we try to do is build those tools that allow you to do rapid generation cycling, which is what they do in Monsanto, which is why they have true breeding strains after a couple of years instead of 20 years, right? So by being able to identify the genes that you're interested in and grow your plant until they're 6 inches tall, because you don't need a flower, right? You don't care what the end result is until you get to the end result. Right, so you do your rapid generation cycling, you can get 5, 6 generations a year because you only need a few hundred seeds to be able to follow the genetics, to be able to make sure that your breeding program is hitting the marks so that you get to the final product with all the genes in one little plant, and then that sucker you grow out. So that can happen in less than 2.5 to 3 years, as opposed to traditionally 12 to 15 years, right? So now we start to look like a big ag, industry. We're using the same tools, and then we go after the same things, patents on those unique varieties, so that we start to look like the people that are going to try to come in and take us over, so they can't just come in and do it. So.
I was going to say I agree 100%. I mean, the days of sharing genetics, is righteous and cool as it was back in the day. It's a recipe for disaster, for the future. I mean, I've had this nightmare for the last 3 or 4 years in Monsanto, is just going to every clone bank in California and picking off every strain. There's a good chance my paranoia might be true.
So, just so you know, I have a great job. People give me free weed and they say, please try this. I'm like, okay. But the other thing I get to do is I get to talk to a lot of people around the world who have big, kind of preservation grows, right? So there's guys out there who, I tell you what, it's good to be rich, because I have spoken to people who have buildings in Colorado or buildings in Chicago where literally it's a 3 or 4 story building and the entire building is dedicated to nothing but, preserving old genetics and seed banks. And I know a guy who's got every seed pack from Sensi Seed for I think it was from 78 to 92. He's got every seed pack. I'm so the issue is that if you have these things, right, and you have no way to get them to the next point, they're not doing you a lot of good. But diversity projects like that are absolutely necessary for the industry, because here's what happened when we went underground. We only looked at one thing, that thing that could get us high. So all of our strains are now very hybridized for one thing, for high THC. We have trouble finding anything that has high CBD unless we go to hemp, right? And so that means that all of the other cannabinoids like THCV and CBC and CBG and CBL, all the things that may be just as medicinally important, are broken. Literally, they are broken. When I look at the genes by a little sequencing DNA, I find big deletions in the other cannabinoid synthase genes. So the only way we're going to get that shit back is if we go to these old preservation grows and these old seed packs, and we start cracking seeds and finding land races and breeding the stuff back in that we lost because the only measure we had for 25 or 30 years underground was the seat of our pants. How high did it make me, right? And not all cannabinoids are designed to do that.
So for those who want to take that initiative and start breeding their own strains, how do they do that?
So, science is your best friend, even though it costs money. And so it basically, if you're a serious, right, so the industry is divided in half. There are, well, not necessarily half, but there are two camps, right? There are the growers and cultivators, all right? And then there's the breeders. So you can be both. You can be a breeder and a cultivator, but you tend to have a mindset or the other. A true breeder cultivator is going to be somebody who is looking to establish a mapping population. You have some donor germplasm that you're interested in for whatever reason. It's got great terps, it's got high CBG, it's got whatever it is. It's disease resistant, it's, it's really great yields, and that forms part of your germplasm. That forms part of your breeding program. Breeding programs cannot be willy-nilly haphazard, right? You have to have a goal, you have to have stock that you can use to get to that goal, and you have to be able to keep really good records, right? Because you have to do reciprocal crosses, then you have to look for the phenotypes, because chances are if you find a phenotype you like from a seed bank, it's an F1 hybrid, and you will lose it for 2 to 3 generations before you find that phenotype again, because it's a hybrid, and when you breed it to anything, even itself, it goes away until you can find it again, right? So that's the reality of plant genetics. And so, you have to keep good records, and you have to be able to find metrics, and so what you do is you essentially build yourself a mapping population. If you have the parents that you reciprocally cross, and you have a couple of crosses, you end up with a germplasm map that, if you can measure it within a couple of crosses, you can build a marker for it, right? So that's the power of organized plant genetics and what you can do if you go about it the right way. You can build a tool that you can use to make money and map anything you want and find it and breed it for future, right? But you have to have a good understanding of plant genetics and how reciprocal crosses work, back crossing, what it takes to get to stabilized strains, and true breeding strains. If you start to understand that, you will build your breeding program so that you can get to these things, and then it's a breeze. Like literally, it's a breeze. If you want to find CPG, we can measure that. You're looking for flowering time, you can measure that, right? So all of these things are observations that you can follow. Good notes, good records, and good germplasm. That's all you really need. So.
And a lot of time.
And a lot of time.
A lot of time, and also, let me put in a plug. I was just complaining before this thing started. We were talking about plant breeding, and I was complaining about, in my lifetime, what's happened to tomatoes and strawberries. They used to be good. They ain't anymore. That's our plant breeders. They knew exactly what they're doing, but they were breeding for something that was bulletproof and could be shipped. I hope you guys that are plant breeders are going to go for not only products that can make you money, but products that the consumers will appreciate. Things that have good flavor, good smell, good whatever. Disease resistance, I think, is terribly important because I don't want a lot of pesticides on my stuff. So
powdery mildew kills more than anything. Powdery mildew is your worst enemy.
Oh yeah.
It kills more than anything else in the cannabis industry, right? So.
And I'm not sure I wanted to smoke neem either. So.
It's interesting because Stefan said something which I've known because I've been in plant biology for a long time, but like tomatoes are engineered to withstand a 13-mile-an-hour impact. Think about that, right? So they're harvested green and thick-skinned because the harvester, when it picks it up and spits it into the catcher, it's a 13-mile-an-hour impact and it can't bruise. So that's what they decided to use their knowledge of genetics for, to make it so you could basically play softball with it, right? So.
And they've done the same sort of thing to grapes. I mean, what's the difference between wine grapes and table grapes? For table grapes, they bred for things that can be shipped. They bred for things that are insanely sweet, but no other flavor other than insanely sweet. And that's basically your high THC with nothing else. So the plant breeders, I want you guys to be artists and go for something good.
So it's interesting because, so some of the things that I think is important for people to understand is, traits. Traits don't exist in a vacuum, right? So it's a physical thing. It's a piece of DNA that has a physical location in the genome, and it may be part of a pathway, right? So when we look at terpenes, we need to start understanding relationships between things like geography and effect. So there's something called, to break out fancy words that don't mean anything, linkage disequilibrium. Basically what it means is the closer two things are on a piece of DNA, the less likely they are to be separated, and the longer that distance is, the more likely they are to be separated, right? So now if you have genes that cluster together, which happens with terpene genes, you'll have 3, 4, 5 terpene genes in a 20,000, or actually that's not true, in a 100,000 base pair section, you'll have 5 or 6 genes. Well, chances are that 100,000 base pairs is not going to get broken up through recombination when you make gametes, that the pollen or the ovule, right? So if you start to understand these things, now genetics and breeding take on a different meaning, right? So let's say you like something from Southeast Asia because it has those 4 terps that make this effect. Well, now you know, because anything from that lineage, you don't need that particular one. You can get that one or that one or that one, because chances are, because they are related through a lineage background, they will carry the same things. So now it becomes an easy thing to start thinking about, how do you maximize a breeding program, right? So, because if you have good representation of the different traits you're interested in, right? It's really easy to do a lot of crazy stuff and come up with some really cool stuff without a lot of work. You don't have to have a repository of 10,000 starting strains if you do it the right way, and you have good representation of those things that you're interested in, right? So.
Yeah, I've always noticed that there, at least for breeders, at least the strains that I've grown and developed, that there's definitely a relationship between structure and terpene profiles. They're probably very closely linked on the DNA, is what I'm guessing. Have you guys had any evidence of looking at structure and terpenes and seeing any relationship?
So, DNA is a weird thing. So it's not, you tend to find, because of evolution, right? So this is going to sound like Bio 101, which is what I'm trying not to do. But because of evolution, what ends up happening is you end up with a gene that, because it does something cool or good or useful, the best way to fuck around with it and make it better is to make copies of it and fuck around with it, right? So we see that in highly responsive systems. So by that I mean that systems that tend to have a direct response to the environment, okay, tend to be highly duplicated and replicated genes that then undergo rapid evolution.
It's like cut and paste.
Right, exactly, it's cut and paste. And so, because Mother Nature doesn't reinvent the wheel, she just takes that and then just modifies what she's already invented. Right? So terpenes and cannabinoid synthetases have very similar motifs because of binding pockets and other things. They use precursors that are actually all built from the same thing. So if you look at the chemistry that we like in cannabis anyway, they all start from a 5-carbon unit called an isoprene unit. Everything in the frickin' world comes from an isoprene unit, including things like plastic and rubber, and you name it. If it comes out of a plant, it starts with this 5-carbon chain, right? So but terpenes and cannabinoids are also built from these 5-carbon chains. So a lot of the enzymes that we are looking at, because they are responsible for making cannabinoids and terpenes, all have similar binding pockets because of the precursors that go into it that get worked on, right? And so what we see is a lot of familial similarity, because you have a need to hold things in a certain juxtaposition to something else to get a chemical reaction to happen, and that motif is just modified. So when we look at terpenes and we just isolate and we look at terpenes, what we see is that there are like 3 or 4 parts of a terpene molecule that are invariant, and everybody has them, and everything else just doesn't fucking matter. And so that's where you get all this diversity for terpenes. And that's why, and it's really weird because terpene genes are very promiscuous, right? So you can have one gene and it makes 4 or 5 different compounds depending on what precursors you feed it, right? And then makes matters worse is there's 2 main precursor pathways. There's the DMAP pathway, and there's the MPEP pathway, and so that gives you GPP or FPP, and then suddenly you're off to the races. And now, you know, you get 30, there's only 30 terpene genes in cannabis, which we found freaking amazing. I'm like, wait a second, that makes no sense. How do you get 200 things out of 30 genes? Well, now we understand why, because depending on what you feed it, at what time of day you feed it, you know, you'll end up with a different set of things that come out of the same genes because they are promiscuous. And a lot of the genes that we see are like that in cannabis, which is very interesting. But now if you think about cannabis, cannabis is one of the only plants that exists everywhere, right? Like there's not a place that you can go you won't find some version of cannabis, right? So, and plants can't move, right? So they have to have all their diversity inside to start with. And that's how they get around doing it. So it's a pretty fascinating thing, and we find stuff that just wows us every single day. But some of it we can turn into products, which helps you guys, so that's cool.
Yeah, but the whole story that you got to think about, your genes and all is what we call in old-time plant biochemistry pools, and pool size. You've got these enzymes that can accept different things, and there are all these intermediates that go to the next guy in the chain. Depending on the growth conditions, depending on the chemistry, all sorts of things, the pool sizes may vary a little bit, and you think you've got all the same genes, but because you have variations in pool sizes because of other things that are happening in the cells, all of a sudden you start kicking out something completely different. And that's something that's really easy to, that's my theory is why tomatoes taste so bad when they were breeding for cardboard and all those other pool sizes that have those nice flavors, it just lost them.
So you mentioned something really good. So it's really important to understand too. So as we mature as an industry, right? So one thing that's going to be absolutely important is there's going to be a split between medicinal and artisanal, recreational connoisseur, right? Because at the end of the day, there are chemicals that are necessary to be medical, and there are other things that are not medical that are effect-based, right? So, or experience-based. And the medical stuff, THC, CBD, that's going to be, you know, like any other commodity, is going to be pushed down. It's going to be a race to the bottom to make the most amount for the least price, right? So that's not the business that most of us want to be in, right? So what we want to be is we want to be recognized for the hard work that we put in and the craft of what we're doing, right? And so what's important to understand there is that growth, or let me say it a different way. There's a biological equation that can never be broken, and it is genetics plus environment equals phenotype, right? And if you keep the genetics constant, but you do not keep the environment constant, your phenotypes will change. We have data from that, right? I can show you where if you use HPS versus LED, you will shift from making more of that to more of that. It happens. I have data from collaborators in Israel where they have the same exact genetics from in 4 different greenhouses in different areas, and the greenhouses were built out of different materials, even though they're all built to the same spec, right? So the glass is different because they got it from another place or whatever, and I can show you how the thickness of the glass or the color of the glass ends up changing the maturation, because when they harvested it all at the same time, X number of days afterwards, the chemical profile on all 4 of them, which were identical genetics, was different. Okay, so all of these things matter. So to be able to get to the top of that craft and to be able to have a product that you can show as your own and then be able to patent, you guys have to understand the basic biological equation, right? Because it makes a difference. So, and that starts with the good genetics. Good genetics, there is such a thing as good genetics, right? Good genetics means that you understand by analysis that you have the highest proportion of good genes, right? So, for instance, we have a patent that we just filed on a SNP that we found that tracks to the highest levels of THC production, right? So you can breed with any donor, but do you want to pass on the potential for levels, or do you want the top levels, right? So So there are good genetics, there are okay genetics, right? And so being able to identify that your strains have the best possible genetics to put into your breeding program is an important aspect to getting it right when you start your breeding program. So.
Yeah, a lot of times you think you have something good if you're doing phenotype hunting, and then you run it and the environment changes and the phenotype changes, and now you're like, well, I made a bad choice. So I think the ones that develop into really kind of like universal strains that a lot of people run are just strains that are consistent in lots of different environments. I mean, I think it's plasticity, is that the term?
It is exact plasticity, and it comes from it being a hybrid. Right? So remember, hybrids, like mutts are hybrids, right? Mutts are great dogs. They're healthy, they don't get hip dysplasia, but at the end of the day, you can't get a purebred out of a mutt, right? So, and that's the kind of thing we're talking about. So, but the good thing about cannabis is you can get a purebred out of a mutt. You take an F1 hybrid, and you can start to sell it. And you can do all these things that help you pick out the pure pheno that you are looking for from that population. So there are things that you can do to make the system work for you.
Yeah, I mean, ideally, at least I'm not a professional breeder by any stretch of the imagination, but from what I've read, the way to go about it is to take inbred lines for many generations and then hybridize them, because you have more stable offspring.
Well, then that's the classic corn model.
Well,
that worked for
but
cannabis.
it's interesting because it's the classic corn model, but it's also the model. It's the model on which all of the patent office stuff is built, right? They assume that when you say F1 hybrid, it comes from the classical definition, which is very well characterized homozygous strains that are then bred together to give you an F1 hybrid that is well characterized, and you can then make certain expectations or certain predictions based on it. Right? So that's what the patent office expects, and that's why we're in the world that we're in right now, because that's not what they freaking got. So.
Yeah, that's what takes lots of time and lots of space.
Right on. So you guys kind of actually answered our last couple of questions. So if there's any questions from the audience, we'll open it up for Q&A.
We kind of like run them up there. Sorry.
No, it's good. That's a lot of useful, resourceful education, and we're all here to learn about genetics, and especially the IP. That's huge. So does anyone have any questions?
Hello, my name is Ben. I'm with OG Cannabis Insurance. I have a question in regards to patents, and maybe it's just a hypothetical question because I don't know if it's played out yet in the court of law. If someone files a patent on a strain and has all the perfect storm, which you guys are talking about, to actually create the strain or to patent the strain and everything else, if they come to find out someone else is using the strain and doing all that, how does that play out? Has it been played out where someone goes after someone else, and how far would it get in court, being that with the federal jurisdictions against it and everything else?
Well, if you'd asked me like last week, it would have said, well, it hasn't happened yet, but somebody just sent me a lawsuit that was filed in Colorado in the last couple of weeks by United Hemp against someone that they say is violating their patent on a high CBD strain. Is in the district court in Colorado. My guess always would have been that, well, the courts, as long as it's illegal federally, are not going to handle it, and people are just getting patents because they figure that this is going to reverse. And I think I'm probably still right, but I don't know. Since now we have a case, we'll have to wait and see what happens.
Well, so it technically can't happen because patent suits occur in the federal circuit, right? So as of right now, you can get a patent because it's not illegal. And by the way, it's never been illegal to get a patent. If they tried to file a cannabis patent in the 1970s, they would have let it happen.
You would have had a great time.
Yeah, but they hid behind the fact that it was illegal to make everybody think you couldn't do it. Right? So as soon as you called and asked, they had to do it. So it's a great question because right now, until it becomes federally legal for a patent suit. Now, the difference is if you can prove that you have a patent and that you have been conducting business where you are selling under license, and you can show that this person has not acquired a license and is not the originator, you have all that stuff in the perfect form, you can then file a civil suit.
That's exactly what's happened in Colorado, but it's in the federal district court. And my guess would have been the judge is just going to dismiss it, but I'm not sure. Let's just wait and see.
Maybe.
My question is, when you guys breed or when you guys play with, I'm assuming that you guys start with cannabis, and do you guys introduce other fees, or is it just within the cannabis?
What you're describing is genetically modified, and so nothing that we've talked about is genetic modification other than accelerated breeding, right? So marker-assisted breeding is regular breeding, just looking at the DNA as your tool, as opposed to growing out the flower and measuring the chemistry afterwards, right? So other organisms or other agricultural, like corn, some corn is genetically modified, some, so that happens.
It's all commercial stuff.
Right, so, and that has not yet happened in cannabis. Nobody has a genetically modified cannabis out yet. However, there are patents on it. Tweed from Canada has a patent for a CRISPR-Cas9 knockout of THC, so that they can make a completely THC devoid plant, right, that makes other things. So, now, in my opinion, because of the way the majority of the cannabis industry is, a genetically modified cannabis plant is not going to be a big seller in the industry. However, that gets back to what I said about how there's going to be a split in the industry, right? So there's going to be the big ag market for mass production of cannabinoids to be put in pills and whatnot.
GMO.
And that'll be all GMO. That'll be all, just like the back of it, all be GMO, and then you'll have the non-GMO artisanal stuff, which we smoke, so.
Yeah, it's really hard to think of a reason to make a GMO artisanal strain. I mean, disease resistance is the only thing that comes to mind. I think there are some things you could probably do that would be acceptable.
You can do that with regular breeding.
Next question.
Hi, I'm Ron with growlands.com. Our platform allows you to kind of keep that acute track of what you're feeding your plants and when you're feeding it. You said something that kind of triggered the nature versus nurture argument. What matters more when you're trying to breed the best possible pheno or cannabis possible? Is the nurture or the nature? I know they both matter, but which one would you say trumps the other?
There is no way to answer that question. So it's the chicken and the egg story on steroids. What happens is you can have, and it depends on how you mean to go about it. If you are looking to breed something from scratch, you're starting with stuff, well then clearly you need the good genetics to be able to get to a place, and then you optimize with the nurture, right? Here's what has happened in the cannabis industry. A lot of people have come across strains, and by sheer dint of luck, have managed to find it and put it in the right place to grow really well. Right? A lot of our classic strains, like Reggie, Kush, Willie Blue, all these things, they were outdoor grown. They found some place where, holy crap, that thing does great. Look at that. And that becomes that family's business for 4 generations. Am I telling you?
Yes,
Very sure. So now, what happens when you try to take that plant and you grow it somewhere else? It doesn't grow as well, right? So that the example I'm trying to start there is sometimes you will find something, and because of where you find it, it's already optimized, and the nurture has already optimized program, you have no way to know that. You're trying to create something new, and in the process of creating something new, you will have to try different nurture to determine the breadth of the expression of that. Because at the end of the day, DNA, unless you have a true breeding strain with 100% homozygosity, which means that you have zero plasticity in the genome, right? Everything's locked. And unless you're in that situation, right, you will have a kind of, you'll be chasing your tail, right? Because as you won't know what the potential is until you've grown it from different things. DNA represents a potential, right? And the nurture represents how that potential is expressed, okay? And so, again, in some cases, you got there because you got lucky and it was there and it was the best it was ever going to be, and anything else made it worse. When you start from scratch, it's the other way, and you got to build it and then try it and then see what happens.
I mean, yeah, this is, I think it's important that if you're growing in a certain region, right, or in a greenhouse or indoor versus outdoor, that you know that the variety that you're growing is bred for that region or that environment. I mean, otherwise, you know, it won't cross over a lot of times.
And that's a great point right there, because now we get back to the part where we're talking about, well, you know, some things travel together, right? So if you find a strain or a couple of related lineages that do really great someplace, now because you know and you understand these two things which are related grow great in this humid environment that's only got that much sun, if that's what you're trying to build, then now you know to look for those things and use those as your starting block, right? So you know, a lot of it is, again, it's, and the power of genetics is about the power of observation. There is not, you can't possibly collect enough information to get to the point where you're useless, realistically, right? Because every one of those things you can observe ultimately has a tie to some gene or some pathway that makes it to that. Right? So everything's useful information, and great notes, great observation skills are what make you guys better cannabis producers.
And the take-home lesson also is if you're breeding, at least I would breed, and I'm not a cannabis breeder, until I know what I'm doing or when I'm working on something, you know, keep the conditions the same each time. Don't go all over the place, because then you're going to get totally mixed up.
Right.
And your record keeping is going to be bumpkus. It's not worth anything.
I couldn't say it any better. So, when you're starting from scratch, the thing that you're trying to create from that's new, that has to be done under consistent conditions. Otherwise, you're going to lose that, and you're going to be chasing your tail, because if it's not done the same way every single time, you've introduced a variable. Remember, like I said, just taking 50-degree water, putting it in a room at 78 degrees at the roots, will completely change the terpene profile for that short period of time. Right? So,
Yeah, even the amount that you water plants will affect it. I mean, there's so many, it's very difficult to do consistent, you know, quality flowers that are always the same, because there's so many variables.
I think Dr. Kirchanski, Stefan, I think my question is more geared towards you. When you discover or unwittingly are promoting a product or something that has patent currently that you're unaware of, like how are you able to detect that before undergoing that? Once you find it, is it your typical cease and desist? Is it what are the procedures?
Well, first of all, we just talked about the fact that they probably can't sue you yet, but let's assume that they can sue you. One of the things about patents is notice that you have to know that there's a patent, and that's why before people send a cease and desist, or even with a cease and desist, they'll send you the patent. And if you honestly don't know about the patent, of course, if this guy is going to advertise his patent everywhere and it's in every single magazine that you pick up, chances are he'll be able to show that you knew about it. But if you don't have notice, you're okay until you get that letter. This is hypothetical because it's not going to happen yet. And at that point, There are some people in the patent world that just want to shut you down, but most people, I think, usually want a piece of the action, and they probably want to try to license the few or something. And
Right.
if you were to get this letter, then you probably would talk to some attorneys to find out whether the patent is good, whether it really applies, whether there's something wrong with this patent. And then if it looks like you're stuck, there's no way to get around the patent, the first thing is to talk to the guy to see if you can come up with some sort of a deal. In a completely different area, paper trails and all things, I had a client that got sued by somebody, and we told him, well, this patent is crap, but an existing U.S. patent is presumed to be valid, so it does have some teeth. And we went round and round and round. It was costing my client a whole bunch of money. He offered to settle with this guy and pay him a royalty and give him $200,000 just to go away. This guy was greedy and said, no way in hell. So we attacked his patent in a patent proceeding, and he shut his patent down, so he got nothing. He could have had $200 grand, just bam, like that. But that's no problem.
Is there a directory, or is it just go in the patent office and look under a specific category to kind of research before?
Well, the patent office has a very good database. I mean, all this stuff that you see in Google's patents and everything, all the different web pages that you're seeing, it's almost all picked up from the patent office database. And maybe it's a little arcane to use it, but it's easy enough to go and look and find out what's there.
Right, yeah.
Use anything but the patent office search engine. Anything
Yeah.
else, it doesn't matter. That's
Give me like three. And give me three besides the patent office.
Google Patents, Free Patents Online, and there's another one that's from me. But the best out there is Google Patents, Free Patents Online.
Okay.
Unless you like torture, in which case go ahead and use the Patent Office.
I'll redirect people to the other ones.
Yeah.
And I sent you a link to the request, so.
Yeah, the only good thing about the patent office thing is if you know what you're doing, you know how to go through the stuff. Google is usually very good, but sometimes they just totally miss it because their algorithm sucks. So you have to know what you're looking for.
My favorite is Free Patents Online. That's what I use.
I agree, everybody. If they all agree, then you know that you got something.
That's the OCD part of every patent attorney and patent agent. We're all OCD, and we all check multiple things to make sure we get the same answer everywhere. If we don't, it keeps us up at night.
We got time for one last question. Do you mind if I ask some questions? Serious questions? Okay, first question. You said that currently people are only breeding and not genetic modifying. Is that due to lack of technical skill or design?
Well, you have to understand the plant before you can modify it, so realistically, there are very few companies out there that have any useful information on the cannabis genome. So without useful targets, you can't really modify anything. So it has more to do with a lack of understanding than anything else.
I think there's certainly a little bit of PR that most of the people that are certainly in artisanal cannabis would not like GMO. So it's probably just bad PR. So it has to do something really great for a strain to make it worth the risk.
You also mentioned that there's some people out there that have archives of previous strains and previous seeds, and they might have medical effects that could be positive for the community. If we don't really have genetic modification and we're just breeding, wouldn't it take years before we see the actual first pharmaceutical cannabis-based drugs?
Well, that's a hard one to answer because if you don't have an analyte to compare it, then you don't know whether or not it will have an effect, right? So, but on the other hand, I think we know that there are, from what we can see already from the strains we have, that there's at least 144 cannabinoids, right? So, I think at that point we need to figure out how many have medical effect already, and then from those, we will be able to do augmentation breeding. Further, with the number of land races still out there and the number of seed packs going back to 1970, whatever, Holland Seed Bank and Cincy Seed, because there are people who have, unbroken seed packs that, would probably viable seeds. I'm pretty sure that we will be able to find anything we need. It's just a matter of looking, right? So, and that's the tough part, because unless you unpack them and start to analyze them, which is illegal, right? So you won't ever find what we have, right? So that's the real underlying problem. Lack of research, lack of unfettered access, and the constant, who's going to pay for it, and will I go to jail for doing it? So, which I honestly, every day of my life since I joined Steep Hill, I look over my shoulder because everything that I do for research breaks every freaking federal law regularly, right? Because you can't look at DNA without making an extract. You can't look at the chemistry without making an extract. So everything that I do to further the industry's knowledge of cannabis puts me in harm's way.